Good morning. Okay, so last time we were talking about mutations and disease, and I want to bring this together with a few more examples. Remember, the gene exists in the chromosome. Mutations in protein coding regions are observable in the DNA sequence. Okay, it's important to remember that the mutation is there in the DNA, but as a result of transcription, processing, and translation, you have a change in the protein, which ultimately affects adversely the action of that protein. So don't think RNA mutation, think DNA mutation. Okay, we will be referring to the genetic code. I'm going to remind you from last time. It's a three-letter code, and we show it in either the DNA form or the RNA form, depending on what nucleotide sequences were given. So remember, it's a three-letter code. For example, the upper left corner of our chart has TTT being phenylalanine, and also in the single-letter code, F. So all 64 possible codons are used, 61 of those to encode amino acids, and 3 to signal stop or termination of the protein. Here's the genetic code in our RNA form, which is the one we'll be using later today. It's the same, it just makes it easier for you not to have to translate nucleotide sequences from DNA to RNA. So you have the substitutions of U's for T's as you would expect. So we're looking at a slide talking about types of coding region mutations, basically what type of change you can get in your protein as a result of particular changes in your nucleotide sequence. So the wild type allele is represented on the top, and the codon in question is a glutamine codon, which is Q. It's a CAG in the wild type sequence. We can have varying mutations or varying effects of mutations on that amino acid. The first one in the second line is a silent mutation because it changes a CAG to a CAA. If you look on your genetic code, you find out that that still encodes glutamine, and so it's a silent mutation, has not changed the amino acid sequence of the protein. Then we have other types of mutations where we do change the actual amino acid. In this case, that C, instead of reading a CAG, now reads AAG, which if we look on our genetic code, is signaling the introduction of the lysine. So that's a single nucleotide, single amino acid change, and that's called a missense mutation because it changes the amino acid from one to another. So this is what I just said in the above. We had a CAG, encodes glutamine. We changed it to a CAA, which is a silent mutation, and then we changed it to AAG, which is a missense mutation. Now those may or may not have serious effects on the protein, probably not. Okay, the other two types of mutations that are shown here are the nonsense mutation, which is our CAG is now a TAG, which encodes a stop codon. Now what that's going to do is basically stop production of that protein at that point, and the rest of the protein after that stop will not be produced. So that's mutation exists at codon 9. That means that the protein that will be produced is only 8 amino acids long. The rest will not be translated. Eukaryotes do not reinitiate translation once it has been stopped on a particular mRNA. So that would be a very severe mutation because the rest of the protein simply will not be made. Okay, the last one is frame shift, and we have to consider what's going on with that. Remember that translation involves reading of the codons by the ribosomes in groups of 3 nucleotides, and that reading frame is set by the AUG. So once we get our AUG, we start translating in groups of 3, and so mutations that change that, delete or add nucleotides, are going to change the nucleotides that are actually being read because they'll shift the reading frame and you will not get the correct amino acids. Well, it's sort of worse than that. If you add or delete particular nucleotides, the ribosome won't know that there's a missing nucleotide and will just continue translating past that point, if it's a deletion or insertion or whatever. So what you end up with is a few wrong amino acids placed into the protein because of the shift in frame. But what's worse is that there will soon be an previously out of frame stop codon that's now brought into frame, and then the protein will stop being made and no further translation of the RNA will occur. Okay, so if you add or delete 1 nucleotide, you get a frame shift. 2 nucleotides, a frame shift. If you add or delete 3, you delete one amino acid from the protein, but it does not actually produce the frame shift. Okay, so here's our sequence finally, where we're looking at the premature termination that results from the frame shift. We've deleted one nucleotide, and that shifts the frame so that instead of having our glutamine, we now have an AGA, which is being read as arginine. And then reading on in groups of 3, we have some incorrect amino acids. But worse, when we get to the TGA near the end of the line, that is a stop codon, and that will terminate the production of the protein at that point. So the original was a CAG. Now we have a deletion of one nucleotide, the C. So now we're reading AGA, CTC, TGG, etc. But then we encounter a stop, and again we terminate the synthesis of that protein, and you will not have further production of that protein. So that would be a very severe mutation in that protein. Okay, this next bit is something that I got from the Genetic Society course source, and it's intended as a sort of in-class activity to basically reinforce what we think we know about, what we do know about the genes and genetic code and the effects of mutations. And I acknowledge the Genetic Society for making this available to us. Okay, so now we're presented with a scenario. We have 2 boys, Liam and Elijah. Liam has muscular dystrophy. His brother Elijah does not have it. Okay, and we're going to compare the sequences of the 2 boys to find out what might be the cause of the disease in Liam. So we sequence the genes from both, and we find 5 nucleotide differences between the 2. Okay, they're listed here. In one case, we have Elijah has an A and Liam has a G, and the location in the gene is the promoter. For mutation 2, we have a UCU codon in exon 19, which is going to now be a UCA, so there's one change. We have a CAA codon in exon 52, which is now a UAA. We have a C to T change in an intron. And finally, we have a GAU to a GAG in one of the exons. So our job is to try to decide which of these mutations is the most likely cause of the disease in Liam. Okay, so what we do, we look at the genetic code for the codon mutations and try to decide what the changes will be in those particular amino acids. So changes number 2, 3, and 5 are both within the open reading frame, and they change codons. So we look at our genetic code and find out what those are. Okay. So we can see that Elijah has a UCU and Liam has a UCA. Well, both of those codons encode serine, so that's a silent mutation, so that's not too likely to be a cause of a disease. The next one is the CAA glutamine codon, which now reads in the disease form UAA, a stop codon. Now that's a nonsense mutation. Finally, we have GAU, which is aspartic acid, and then GAG, glutamic acid. So that's a change in the amino acid, and it's a missense mutation. So of these, which do we think is the most likely to be the cause of the disease? Mutation 2, the silent, mutation 3, the nonsense, mutation 5, the missense. Well, yes, of course it's P. Okay. So here's an interesting question. What would be the effect of mutation 3, the stop codon, on DNA replication? No effect, replication will stop. Okay, so the answer is A, there'll be no effect. Right? People get confused about this sometimes, that the stop codon refers to what happens during translation, and it does not have any effect on DNA replication. DNA polymerases don't read codons, they just copy nucleotides. So there's not going to be any effect of this on replication. Similarly, what will be the effect of mutation 3, the stop codon, on transcription? No effect, transcription will stop. No effect. Same comment as before. Transcription is done by RNA polymerase, which does not read codons. It starts and stops because of sequences of DNA sequences in the promoter and terminator. It does not read codons. What will be the effect of mutation 3 on translation? No effect. Translation will stop. Well, of course, translation will stop. The stop codon refers to translation and involves ribosomes. So the stop codons lead to release of the new protein from the ribosome and the end of translation. but has no effect on transcription or DNA replication. This is an important point. So now we get to look at a few other mutations. Remember, we have a promoter mutation, an A to a G, and we also have one within the intron. Okay, so which is theoretical? Which of the following types of promoter mutations could affect the expression, to say transcription, of this gene? Data box, proximal promoter element, deletion of 200 base pairs starting at the transcription start, all of the above. Okay, so now I want to move on to our little Betaglobin worksheet, which is the same one that we had last time. It's a sequence of a cDNA that was produced, of course, from a mature mRNA that encodes the hemoglobin beta chain protein. And we're told that the ORF, which is to say ATG, starts at nucleotide 51 and ends at 494. So the ATG starts the protein, and the stop codon, which is the TAA or UAA, ends at 494. So you're supposed to find these in the sequence and circle them to understand what the length of this reading frame looks like in terms of the DNA sequence. So here's our slide. I've got the ATG italicized in capital letters, and the UAA or TAA is also italicized and in capital letters. So this is partly to remind you that mature mRNAs do have long upstream leader sequences, 5' UTRs, and also they can have very long 3' trailer or 3' untranslated regions as well. The open reading frame only is part of the mRNA, the obviously important part, but the other parts are important too. Okay, so here's another practice question. What are the effects of deleting nucleotide 60 from the cDNA sequence shown? So here's our slide. This is the C we're talking about, and we're going to delete that C from the DNA, which eventually ends up as part of the mRNA, etc. Okay, so that's a pretty quick stop codon here. That C, when we deleted it, immediately brings that next codon, TGA, into frame, and that will stop the production of that protein. So here we're showing a non-template strand of DNA, and the codons are being read as shown. We've got the normal one, which has a CGA codon, and then underneath that we have a CAA codon. So there's obviously been a new change, a mutation in that codon, and that could or could not change the actual meaning of the codon. Okay, so you can look at this and say, okay, the mutation shown is best described as frameshift, missense, or nonsense. Well, you need the genetic code to say what the change actually is, but it's a missense mutation because it's actually going to change. It's going to change the actual amino acid sequence. Now, if I'd put silent as one of the options, you would have had to go and look at the genetic code and see whether it was really missense or actually nonsense. Okay, so it's a B missense, right? I looked at the genetic code, and it wasn't arginine, and now it's a glutamine. So it is, in fact, a missense mutation. All right, so things you should be able to do with DNA and RNA sequences. You should be able to look at a cDNA, identify the ATG, or if I identify it for you, be able to read the genetic code, what the amino acids are going to be, for example. Look at the 5' and 3' UTRs, and predict the results of various types of single nucleotide changes that you might have. So this is all practice for things you're going to need to do at some point. Okay, so I'm going to move on to talk about inborn errors in metabolism, or to say some of the large number of genetic diseases that result from the deficiencies in particular enzymes or their inability to do their reactions in cells. Okay, so one general rule about these things is that particular compounds might accumulate to toxic levels and others will be in short supply if you disrupt the function of a biochemical pathway as a result of inactivating one of the enzymes in that process. So our schematic here, again, we've seen this one before. In the normal form of the gene, you have a wild-type sequence in the chromosome. You have transcription, processing, translation, with the result that you produce a protein that catalyzes a reaction in a cell. In this case, it's going to take a colorless substrate and convert it into a colored product, which leads to a pigmented cell. In the absence of that enzyme, if we have a mutation in the gene, which is transmitted through transcription and translation, gives an altered amino acid, If it's at the wrong spot in the protein will not be able to catalyze the reaction, and you will end up with an unpigmented cell as the phenotype. So this is a paradigm for many different types of genetic diseases. You get phenotypes that result from failure of a particular enzyme to actually work. Okay, so our example is going to be phenylalanine hydroxylase, which is located, the gene is located on chromosome 12, and the mutations in this gene cause a very serious genetic disease, that of phenylketonuria, which is abbreviated PKU. So untreated, this is a very severe disease that causes severe intellectual disability, but fortunately all newborns are tested in statewide and nationwide newborn screening programs, and the therapeutic interventions, if they're started early enough, can largely prevent disease progression. So this is a good genetic disease in the sense there is actually something you can do about it therapeutically, although doing so is really very difficult for people, as you will see in a minute. Okay, so here's our chromosome 12. The gene encoding the phenylalanine hydroxylase enzyme is on the Q arm on this chromosome. Okay, just jumping ahead a little or just moving aside a little maybe. Remember that you've got one allele from mom and one from dad, and these occupy equivalent locations on those two chromosomes 12. And jumping ahead also, there are a lot of different disease-causing mutations in this gene in the human population, but they are generally recessive. So you could be homozygous for wild type, two good alleles, heterozygous for mutant alleles, or homozygous for mutant alleles, not necessarily the same as each other. Okay, it's generally recessive, and we can use our normal big A, big A, big A, little a nomenclature to describe families which have this disease segregating in them. Okay, so to get the disease, a child must get a defective allele from each of the two parents. Both parents must be carriers. Okay, so here's the reaction. It looks like a harmless reaction enough. We're converting one amino acid to another. It's hard to believe that this could create such a debilitating disease if it's not working, but as it happens, it does. So our reaction is we take the amino acid phenylalanine, and the enzyme phenylalanine hydroxylase converts the phenylalanine to tyrosine. Seems harmless enough. But you have to consider the broader pathway in which this particular reaction occurs. We're shown at the top, we've got phenylalanine, or amino acids, really derived largely from dietary protein. And the proteins are broken down into the constituent amino acids for uses by our cells or our bodies to make our own proteins and sometimes to contribute to metabolism, etc. So the dietary protein is the source of phenylalanine. And if there's excess, we can have a problem with accumulating of toxic metabolites. I'll get to that in a second. Okay, so the idea is in pathways, you generally have a precursor, a series of intermediates that are acted upon by enzymes in sequence. So in this particular theoretical example, we have gene A encodes enzyme A, and its job is to convert the precursor to an intermediate. Then we have enzyme B, gene B, enzyme B, takes that intermediate and converts it to the next one. And again, a gene C encodes enzyme C, and that produces the product. So the sort of byproduct of this kind of metabolic organization is if you block your pathway at any step, precursors or intermediates that are before that or upstream of it will accumulate, and you could have deficiencies in products because you can't make them because you can't get yourself all the way through the pathway. Right. So in this example, if you intermediate A will accumulate rather if you don't have enzyme B, which if can't act on it. So depending on what step is blocked, you can accumulate different compounds or be short of other compounds. Here's our phenylalanine pathway again, one version of it. Okay, so what's supposed to happen here is that we take our amino acids, including phenylalanine, from our dietary protein. We're supposed to convert them to tyrosine for further degradation through this pathway, which is basically leads to excretion and just further metabolism. So we're blocking this pathway above tyrosine, which would mean that the other steps in the pathway don't happen, which is not actually a problem in this case. What is the problem is that phenylalanine itself will accumulate, as well rather unusual metabolites of phenylalanine, including phenylpyruvic acid and some other ones. So in this case, our pathway is blocked, and that leads to accumulation of unwanted metabolites, which turns out are toxic in high concentration. This is very debilitating to the developing nervous system. So phenylalanine accumulates. Phenylalanine is not converted to tyrosine, and some of the excess phenylalanine is converted to other toxic metabolites. So tyrosine, it seems like, might be in short supply, but we also get some tyrosine from our diets. So that's not really a problem. It's the inability to get rid of the extra phenylalanine that is the problem. Okay. On the other hand, some people with PKU are quite pale because tyrosine actually is used for the production of melanin, as we'll see on a different day. So you may have a little less melanin than you should have because of the lack of tyrosine. Here's another representation of this pathway. The dietary protein is broken down to these and other amino acids, of course. And in the absence of phenylalanine tyrosine, you get the disease phenylketonuria because you can't convert your phenylalanine to tyrosine. Instead, you get accumulation of toxic metabolites. Tyrosine itself, we'll talk about some other day, that itself is used for synthesis of numerous things besides just proteins. Okay, PKU detection in newborns. So preventing the effects of the excess phenylalanine depends on a very, very, very strict diet. So all newborns are screened for high blood phenylalanine levels, among other genetic disease metabolites. The original one, and still the most traditional and cheapest, is the Guthrie test. So what happens is a drop of blood is collected from the baby's heel, and the sample is sent and tested for high phenylalanine, as well as another almost 30 other biochemical tests to detect various other metabolic issues in a baby. Okay, so here's a picture of the baby having the heel prick. The blood is taken out of the heel, and then it's spotted on a card, and it's sent away to the diagnostic testing lab, which looks for excess metabolites or any other sorts of issues, including high phenylalanine. So PKU, if it's detected, the child must be put on a PKU diet that must be maintained at least until adolescence and preferably throughout life. Now this is a tough one on the family. Okay, some more tests are done to make sure the kid really does have phenylalanine, and the low phenylalanine diet must be maintained to prevent the intellectual disabilities that would result from the extra phenylalanine should it be allowed to accumulate. Okay, this is where it gets tough. High phenylalanine foods are basically foods that are naturally good sources of protein. This includes fish, meat, dairy, wheat, eggs, nuts and legumes, etc. Low phenylalanine foods, which do have some protein but are relatively low in protein, include most vegetables, most fruits, and now there are special formulas that are made for PKU diets for people that really can't eat normal protein. So no natural sources of protein are really allowed. So the formula has to be consumed several times a day in order to maintain sufficient phenylalanine for normal protein synthesis, but not enough that you get a huge excess to lead to the debilitating disease. So also diet food containing aspartame does contain phenylalanine, and that must not be consumed. So once a child is an adult, it used to be thought that they could go off the diet. In fact, they can go off it to a certain extent, but it is recommended that phenylalanine levels be kept as low as possible. Where this is really a problem is a woman who's got PKU that wants to get pregnant because she needs to keep her phenylalanine levels extremely low because that would have devastating effects on the developing fetus. So maternal PKU refers to the fact that a mom who has PKU can have a child who does not themselves have PKU, but is exposed to high phenylalanine in utero. And this has devastating effects on the baby. So women who, with PKU want to become pregnant should monitor phenylalanine levels extremely carefully. Here's a clicker question that we didn't do, but I'll just go over it now. People who have PKU must avoid consuming blank to avoid building up too much blank in their bodies. Okay, so the answer is D. Avoid consuming phenylalanine to avoid too much phenylalanine and phenylpyruvic acid being built up in their bodies.